Aloha and welcome everybody to part 2, Ancient Genomics. We will be opening up in just a moment, starting with Dr. Emmet Aluli.
E ho mai ka iki mai luna mai e o na mea huna no e au o na mele e ho mai e ho mai e ho mai na amakua mai ka lihiki a kalakau mai ka ho'okui a ka halapai. Nā aumako I a kahina kua, I a kahina alo, I a akau I ka lani. O kiha I ka lani, ue I ka lani, nunulu I ka lani, ka holu I ka lani. I a ka pulapula a o kou o Kauka hui. E malama o kou I a mako, e ulu I ka lani, e ulu I ka honua, e ulu I ka pai aina o Hawaii. E ho mai ka ike, e ho mai I kaika, e ho mai ke akamai, e ho mai ka mapopona, e ho mai ka iki papa lua, e ho mai ka mana. A ama ua noa.
Aloha and welcome to part 2 of the Hawaiian Genome series. Today's episode is Ancient Genomics, featuring Dr. Keolu Fox and Dr. Leah Dowsett. And just a couple of housekeeping announcements, if you will, please. This is being recorded. This presentation is also being live streamed on Facebook for those that are not on Zoom. It's a webinar, so you won't be able to see each other, but you will be able to see the panelists here. But the panelists will be able to see who is listening in on the call through Zoom. So we are grateful to all of you who are here. You can leave at any time. Yes, we can see all the panelists here. You guys are looking really good. And without further delay, I'm going to turn it over to the moderators for tonight's presentation, Dr. Noah Emmet Aluli and JoAnn Tsark.
Well, aloha mai. Welcome to part 2 of a 3-part webinar series on Hawaiian genomics.
JoAnn, you're muted.
There we go. Is that better?
Yeah.
Okay, great. We had such a good reception from part 1. Mahalo to Dr. Leah Dowsett and Dr. Keolu Fox. The evaluations really confirmed that people received the information and had a lot of questions, which I know our speakers are going to address some of them in their presentations this evening, and had just great feedback for all of us on that. So thank you for that. And many of you said, a good 25 of you said, we're coming in for part 2. So welcome back. For those of you who are here for the first time, part 1 was recorded as well. And the link will be provided for you so you can see it. And we strongly recommend that you look at that. You know, Dr. Dowsett did such a great overview of Genomics 101, which just kind of helps lay folks like myself just understand a little bit more about the science. And Dr. Fox was great about looking at some of the historical studies that we need to pay attention to. That's kind of our learning curve and also raises the kinds of issues, not just in Native Hawaiian communities, but other indigenous communities, of what we need to pay attention to, what we need to be informed about, and looking forward, how this science can in fact benefit us. So Emmet, over to you.
Yeah, thank you. Well, you know, I was really pleased to see the participation of many of our Kauka. The Ahahui on Kauka considers this to be a priority issue. And mahalo, Dr. Leah Dowsett, for a clear overview of the genomics and its application in medical care. Your reference to the Pa'o-Kalani Declaration, highlighting concerns in the Hawaiian community about bioprospecting and biocolonialism. And Dr. Keolu Fox provided a wealth of historic information on genomic scientific studies and cautionary accounts of ethical misconduct, the lack of inclusion of us indigenous peoples, and the growing profits being made from the sale of biospecimens and the sale of access to biospecimens. Back to you, JoAnn.
Yeah, I want to remind everybody that Papa Ola Lōkahi has posted a link that includes not just the recordings for part 1, it will include tonight's recording as well as part 3 when that happens. And they also have in that cache a number of scientific publications and journals so that you can kind of get caught up and look at the body of literature that are being contributed to largely by Dr. Fox and also some of his indigenous colleagues in Aotearoa and American Indian communities about this topic. So I'm going to start with introductions. I don't want to take up the time for our speakers because they have such great things to do. But Kim, if you would put up the slide for the short bios. Great. So I'm going to introduce Dr. Leah Dowsett. You can read the formal part, but you know, local girl, graduate of Kamehameha School. Her family hails from Kauai, attended college on the continent, University of Southern California, where Smarty Pants earned 2 degrees in biological sciences and psychology. Yay. So nice to have our own experts in our Kanaka community. She's grateful to be able to come home and through the Ola program and get her MD from the Johnny Burns School of Medicine. So today a board certified pediatrician and completed her residency at Kapiolani, which she informed me was where she was born. She's also board certified in medical genetics and genomics, and she completed her fellowship training in Philadelphia. So I know Leah's father, Dr. Jerry Wang, and I always give a shout out to him because he was so generous and helped us as a talented biostatistician in our learning curve in community-based research. So he really lent his expertise to us. Leah is passionate about ensuring that science is objective so that the existing barriers to health are removed. and that there is equitable distribution for advances in medicine. She dances hula with Halau Hula Ka No'eau and their kumu Michael Pili Pang, and her family calls He'ea home, where she and her husband is raising their family, a new baby, Li'i, with their two pua'a, mana, and pua. Okay, Emmet, over to you. You need to unmute, Emmet. There you go.
Introducing Dr. Keolu Fox. His research program is multidisciplinary in nature, reflecting his interdisciplinary research experience in anthropology, genomics, and computer science. Dr. Fox explores the links between human genetic variation and disease in underrepresented population. He's got a postdoctoral research at University of California, San Diego School of Medicine. He advocates for more inclusive and representative genome sequencing to allow indigenous populations to gather and analyze their own genetic data. Keolu's current project focuses on using genome editing technologies to investigate the molecular events involved in chronic inflammatory states resulting in obesity and catecholamic resistance. We know Dr. Fox's family. His aunt Leslie Fox was the very first Native Hawaiian nurse on Molokai. His cousin, Dr. Keolu Fox, as we all know very well, has published anything from Kani and Wahine Health and Mana, and is now at Aloha Care as the representative of the Native Hawaiian programs and needs. So, mahalo.
Mahalo, Emmet. So as we go into the presentation part, I just want to open with this quote from a current publication by Dr. Fox and his colleagues listed here below, Kathy Sosi and others. And I think it's a great introduction to where we are, and that is for decades, quote, for decades, scientists have collected genomic information from indigenous peoples and their ancestors with the goal of elucidating human migration events, understanding ancestral origins, and identifying ancestral variants contributing to disease. However, such studies may not have offered much benefit to the indigenous groups who contributed DNA, and many have instead perpetuated stereotypes and other harms. With recent advances in genomic technology facilitating the study of both ancient and present-day DNA, researchers in indigenous communities have new opportunities to begin collaboratively addressing important questions about human health and history. Yet, while there are increased efforts to ethically engage indigenous communities, more work is still needed as the discipline struggles to absolve itself from the racialized science and extra-active biocolonialism that defined its past. And I like quoting this because it really does introduce what we're going to hear tonight. We've asked Leah to begin this session. She's going to give us, again, another brief overview of genomics, just so that we can get on board, especially for those of you who are joining us for the first time. So without further ado, Dr. Leah Dowsett.
Sorry, just unmuting myself and getting my screen going. And then let me start my slides. Can you guys see, hopefully? Getting some nods. Wonderful. All right, so today we're talking about ancient genomics. And again, thank you so much for your kind words, Dr. Aluli and JoAnn. Again, I'd like to acknowledge, our past, current, and future sponsors. So mahalo nui for having both Keolu and myself. Like previously mentioned, there is a more in-depth review of genomics. So you guys are going to get the 2-minute version today. But what is genetics? So genetics is the study of how traits, things like hair color, eye color, or even disease risk are passed on or inherited from parents to their children. Genetics can influence how these traits can be different from person to person as well. So your genetic information is called your genome, and your genome was made out of DNA. And DNA is found in almost every cell in your body. If a cell's DNA is mutated An abnormal protein might be produced, and that can disrupt the body's usual processes, which can lead to a downstream event such as a disease or cancer. So DNA, right, contains all the information that we need to build the entire human body. It's got its own 4-letter language, and it's the order of these letters that ends up determining the meaning of the information that's encoded. So just as the letters in a word, right, the order certainly matters to help elucidate meaning. Same thing with DNA. So genes direct the production of proteins. And so it's estimated that we have over 20,000 genes in our bodies. We have 2 copies of every gene because we inherit 1 copy of our genes from our mother and the 2nd copy from our father. A gene can be as short as a few hundred base pairs or as long as many thousands. And in a coming up example, I'm going to talk about the BRCA1 gene. And that, for instance, it's pretty huge. It's over 81,000 letters long. So genes are packaged into these things called chromosomes, and a person typically has 23 pairs or 46 in total. And again, this entire collection of DNA is known as your genome. So we like to use this analogy, which is the cookbook analogy, right? So taking your genome as the cookbook itself, the book can be organized into separate chapters, and those are kind of like what your chromosomes represent. And within each chapter, you have individual recipes, which are the genes. Going down to a sequencing level, sometimes, you can have mistakes or variations or mutations or whatever you want to call them, but, it could be as big as missing an entire page. Sometimes you're missing a couple pages. It could be a paragraph that isn't right. Sometimes it's as small as missing a sentence or even maybe mixing up a word in the ingredient list in your recipe that could lead to disease. So in terms of genetic testing, I wanted to actually talk a little bit about clinical genetic testing. That type of testing, again, looks for changes. Sometimes they're referred to as mutations or variants in the DNA. And it's useful in many areas of medicine because it helps change the way we provide medical care to the patient and your family as well. Sometimes it can diagnose a specific condition like Marfan syndrome, as an example, or maybe inform you about your risk to develop breast cancer. There are many different kinds of genetic tests. And so, a big part of what we do in my office is kind of making sure that the right person is getting the right test and that we help order those tests, interpret those, and explain the results in a meaningful way. So for molecular tests, which is the main method of genetic testing that we perform, we're basically looking at genes, right? Those instructions that are basically coding for different proteins, and we're looking for variations that could lead to a genetic disorder. We also look at chromosome tests, for people who have extra or missing chromosome, grossly or missing pieces. That's another thing that we do. And of course, sometimes we use biochemical tests, right? Tests from the blood or the shishi to help kind of look for changes that might indicate that we may need to do studies of the DNA afterwards to find a disease. As an example from the last time, I wanted to go over 23andMe one more time. Again, you guys are familiar that this is a direct-to-consumer company that does ancestry testing, but as a part of their package, they do offer a health screening. And so I want to hone in on the breast cancer screening specifically, because that test or screening through BRCA for that company, 23andMe, you're really only looking at 3 spots in the gene. So the 3 mutations that are the most common in people of Ashkenazi Jewish descent are looked at. And again, these are not the most common mutations in the general population. So we know that there are thousands of different spelling mistakes in that gene or in that instruction that can lead to breast cancer. But again, this test, when we're thinking about the beads on the string theory, there are over 80,000 beads on this string, and we're really only looking at 3 of them. So when we have people coming into our office saying, I had the 23andMe done for ancestry, I did the health screening, and I'm not at risk for breast cancer, I think that's a little bit dangerous. And this misinformation can be falsely reassuring. So a lot of what I do, again, is clinical genetic testing, but I wanted to contrast that to ancestry testing because we'll talk a lot about ancestry in a little bit. And again, for genetic ancestry testing, kind of folded into genetic genealogy, it's a way for people who are interested in family history, right, to go beyond what they can learn from relatives or from historical documentation and kind of examine the DNA more closely to give us clues about where their ancestors may have come from, and also maybe elucidate some more information about relationships between members of a family. Again, the more closely two individuals are related, the more patterns of variation that they typically share. So a little caveat, there are limitations to different types of ancestry testing, and ancestry tests really aren't 100% accurate or correct. Some of the limitations include, looking at the database, right? So in order for your ancestry to be correctly assigned, your ancestors, let's say people, who are Kanaka need to be represented in the database in order for the company to use it as a reference. So they can't, tell you that you're somebody from Polynesia if they don't have Polynesians in their database. Another caveat is that in terms of, oversimplifying a lot of populational studies, but some populations share a lot of ancestry. So, here in Polynesia, right, we know that there's a lot of migration back and forth between, Hawaii and Tahiti and Aotearoa. And so when there's a lot of shared ancestry, it's hard to distinguish, exactly, how different groups may be separated. And so looking again at databases helps us kind of refine those issues. I think people who also have a small percentage of ancestry that they're interested in, finding, we can't really be that confident about the results. So like, if you, have a small proportion of, indigenous ancestry, for example, you get, a baby toe worth of, Hawaiian blood per se, then testing is also less accurate. So this is a photo, and it's from National Geographic's Genographic project. And I chose this specific picture for a couple of reasons. So first of all, these 6 individuals, they have the same, racial heritage, if you will. They all did the same over-the-counter ancestry tests, and they were found to have the same amount or dosage of white, sub-Saharan African, and Asian in their genome. I wanted this to be thought-provoking, and I want you guys to kind of take a look and tell me what you think. Some of you in this audience may look at these 6 individuals and say, wow, there's just such great variability amongst these people. Others in the same audience might look at them and say, geez, wow, they look like a family. And some of you might even be going, wow, they look like my family. The reason why I chose this project is because the Genographic project, again, is through National Geographic, and they described this as an anonymous, non-medical, non-profit research initiative that was supposed to use genetics and computational technologies to basically look at historical patterns in DNA from people around the world to better understand our shared genetic roots. Now, on the flip side of that coin, right, if you are certainly more concerned with things such as indigenous data sovereignty, you might say that the Genographic project was actually exploitative and unethical because it uses indigenous peoples as subjects for scientific curiosity and research that provides no real benefit to the indigenous peoples and could put them at significant risks. And the researchers would then take blood and other bodily tissue samples for their own use in order to further their own speculative theories about human history. So just something to think about. When we're talking about genetic genealogy, right, we're talking about using DNA, and there's many different types of DNA that's being looked at in these types of tests. So autosomal tests, right, so this is talking about the admixture analysis where genealogists kind of use this type of test because when we talk about just Y chromosome and mitochondrial DNA, they represent single ancestral lines, while this autosomal, right, your chromosomes numbered 1 through 22, that set does not necessarily capture the overall ethnic background of an individual as well. So using the autosomal, you kind of get a better distribution and a better picture. And then we'll go over a little bit about the phylogenetic analyses. So haplogroups are basically a genetic population group of people that share a common ancestor either on their paternal or maternal line. And again, depending if it's Y DNA, it follows a male descendancy versus mitochondrial, which is a female descendancy. I want to make a note that all haplogroups pretty much started from one original haplogroup in Africa, and then as time and different centuries rolled into millennia, as they've passed, more and more groups have come to be because as DNA mutated each time, different groups have split off and become their own haplogroup. So really briefly, mitochondrial DNA is generally kind of only inherited from your mother. And the reason is because, again, mitochondria, which are these specialized cells in our bodies, have their own genomic information, and individuals inherit their cytoplasm kind of exclusively from their mothers, because when the egg and sperm come together at that instant, the sperm is really tiny in comparison to the egg, and they really only give like the DNA portion, but all the other stuff, that mitochondrial information, really comes from the mom. So it's for that reason that we can kind of follow maternal haplogroups through time. And again, it's useful for genealogy because it preserves information about female ancestors, that may have been lost from historical record because of surnames and how those are traditionally, follow male lines and not passed down in some cultures, for example. This is just a map kind of showing how, again, we know that going back through time, there was a mitochondrial Eve right out of Africa. All humans kind of diverged from, this one original mother, and then became spread across the world. Paternal haplogroups are a little bit more difficult to understand. Basically, it's relating to the Y chromosome. And so that's the sex determining chromosome for men. So basically it's passed down from father to son. And I guess the point I want you guys to take away from this is that it's often used to investigate questions about family related to surname for ancestry testing as well. And so this is kind of a summary of that. So we all have variations in our DNA, and I think it's important for us to focus on what makes us different from each other, right? We have a lot of similarities. So these two species, their genetic code is 99% identical. So delving more into ancestry and, of course, human genetics, it's the differences that make us unique. And again, it's those small differences in really critical areas that can sometimes lead to disease. I want to give big mahalos to Professor Fox, because he has done so much in this area of archaeology and genomics. And so he actually shared his mana'o with me. And so, these slides are based off of his work. So thank you very much for allowing me to talk a little bit about Pacific settlement. And there are many different lines of evidence that tell us about the settlement of the Pacific. And of course, we're going to go over some genetic examples, but I want to talk about some archaeologic ones too. So hopefully you guys are familiar with Lapita culture, right? And it's the name given to the prehistoric people from the Pacific Ocean who left evidence, throughout the islands of the Pacific through different ceramic constructs that were kind of left behind that archaeologists, believe help us map historic cultures. And so this Lapita pottery was found in Fiji, and of course in Tonga, and also in Samoa. So again, recognizing that, some of the population of the Pacific came, right, through travels from Melanesia and other parts west of us. Experts think that Tonga was the first place to be colonized and that Aotearoa was the last. And looking at this specific settlement in Tonga and doing some archaeologic investigations, they were able to kind of date it at around almost 3,000 years old. They looked at different things and tools. And so while today in Tonga, modern artisans use different types of sanders and adzes, there were coral and different types of braiders that they would also use that kind of help trace the lineage back through time. Throughout the Pacific, again, lots of back and forth travel, which we all know, but the last Polynesian settlement was thought to be in New Zealand at Wairoa. And here we look at a paper that talks about looking at the mitochondrial genome. And specifically, they're using genomics to look at maternal haplogroups, right, in mitochondrial DNA, to learn more about the indigenous Maori people there at Wairoa as well. On a larger scale, we can learn more through genomics about our personal selves. So Keolu was kind enough to share this, and this kind of breaks down his maternal haplogroup, which kind of locates him to the Polynesian population, right? So mahalo, Keolu, for allowing us to share that today. But it's also through different types of combined genetic ancestry tests that many people can use, especially scientists, to kind of explore the history of populations, how they arose and how they migrated and then eventually mixed with other groups. So we know that here in Hawaii, the islands were first settled as early as the year 400, and Polynesians, came from the Marquesas and traveled to the big island, of Hawaii in Va'a There was a lot of back and forth, and at the close of the second migratory period, there was about a 600-year gap of isolation until, we had contact again from Western civilization. During that 600-year migratory break, more or less, that's when the foundation and expansion of the classic Hawaiian civilization thrived. But in 1778, that's when the first Europeans set foot in Hawaii. And so when Captain Cook arrived, it was estimated that there were over, around 700,000 kanaka maoli. He brought disease with him, and since then the death rate amongst our indigenous peoples has accelerated after contact. Within 2 years of his arrival, they say that 1 in every 17 Native Hawaiians died. And then over the years, there are many other infections, different types of infectious disease, things like measles, the chickenpox, polio, tuberculosis, maipake. They killed thousands of Hawaiians as well. By 1800, that declined to about 48%. By 1820, a declination down to 71%, even further to 84% in 1840. And of course, after the Spanish influenza, in 1920, the Native Hawaiian population was estimated at around 24,000. So kind of thinking about this crisis, right? And the sense of urgency, there have been many inquiries into the origins of Hawaiian people, though not always performed by Hawaiian people. So this is a 2012 study that kind of looks at the ancestral origins of Polynesians, and they found that a significant portion of our genome does have Melanesian admixture. And again, through some more genetic works looking at DNA and genomes, scientists can actually find that there is links between between Native Americans and Polynesians that predate the settlement of Rapa Nui. They looked at variations in the genome between individuals across Polynesia, and they looked for specific markers that were unique to Native American admixture, and they were actually able to conclude that there was prehistoric contact of Polynesians. But again, all this is to say that this is, again, a lot of fancy science, which is great, and it's a great, fancy study, but also too, our mo'olelo tells us the same thing, right? We as Hawaiians, we have been traveling back and forth throughout the Pacific and throughout the Polynesia basically for centuries, and I think we all know that too. What I'm going to ask Keolu to do now as we kind of turn over is to kind of put it all together and maybe focus more on how we can maybe think more about predatory access to our genomes, and again through indigenous populations, and how we can maybe think about that as a group at large. So let me see if I can stop the share screen. And then Keolu.
Hanaho. No.
Oh,
No, I'm just trying. That was awesome. Amazing. Yeah.
No, thank you.
Shall I,
it's all your work.
Oh, please. Let me share. Oh, I need to get abled. I need to be able to do the sharing. It says I'm host disabled participant screen sharing. So, oh, okay, Raja. All right, perfect. Okay, let me begin. Okay, mahalo everybody. Welcome. I mean, it's just great to be back again with our second installment, and what a lovely series of introductions and just the whole story and all of the data and the presentation that was just presented is amazing and sets this up very nicely. So continuing with safeguarding indigenous genomics and access, I'd like to just focus on some of the points of access where people are kind of negotiating access to DNA, whether it's contemporary, modern indigenous DNA or ancient DNA. And today's talk, we're probably mostly going to focus on access to ancient DNA. So I wanted to start off with something a little bit personal and historical. So I've grown up all over the world, not just Hawaii. I have lived a lot on the East Coast as well. And one of the places I went, because it was absolutely free all the time, was the Smithsonian Natural History Museum. Because this is growing up with my mom, just my mom, single mom. This was one of the only things that we could afford. So it really formed the way that I think about history. And it's a really complex place. You know, I think for children, it's pretty amazing and wondrous. But once you start to creep in and understand the behind the scenes access to a lot of this stuff, it kind of changes the way you think about museums broadly. Now, this one is supposed to be free. It is free, but we also pay for that with our tax-paying dollars as a part of the United States of America. And as a kid, I always wanted to be Indiana Jones. I remember dressing up as Indiana Jones for Halloween's, which is coming up around the corner. It's kind of strange because it takes a while until you kind of put it together that what this guy is doing in this picture right here, Harrison Ford, it's wrong. So it changed my opinion about a lot of these things because I began to think and learn more and more about history and who gets to tell history and how those artifacts are interpreted and what the profound responsibility is. And so I think the more and more you learn about the history of colonialism and imperialism, you realize that history isn't as objective as you think. But this individual, the guy, Indiana Jones, that Harrison Ford plays, is actually based off of this man, and his name is Hiram Bingham III. Now, what's interesting about this individual is that he actually grew up in Hawaii. He was the son of missionaries, and he would later go on to become one of the most famous archaeologists in history. Now, he is one of the most famous looters, grave robbers, and tomb raiders in history, coming out of the kind of 1914, 1920s traditions of the way that people did archaeology. And this is actually the character that Harrison Ford is based off of, hat and all, as you can see in this photo. Now, that might explain why there are tons and tons of artifacts from all over the world in museums and warehouses that are stored in places like the Smithsonian and the Natural History Museum in New York City and many other places. And I think something that illustrates this really well is this short video clip that I wanted to share with you guys. And I hope it works. And I think this explains where a lot of this stuff came from. It's from a recent movie called Black Panther. So I'll play it now and you guys can have a look.
Good morning. How can I help you?
I'm just checking out these artifacts. You tell me you're the expert.
You could say that.
They're beautiful. Where's this one from?
The Bobo Ashanti tribe, present-day Ghana, 19th century.
For real? And what about this one?
One's from the Edo people of Benin, 16th century.
Now, tell me about this one.
Also from Benin, 7th century. Fula tribe, I believe.
Nah.
I beg your pardon.
It was taken by British soldiers in Benin, but it's from Wakanda, and it's made out of vibranium. Don't trip. I'm gonna take it off your hands for you.
These items aren't for sale.
How do you think your ancestors got these? You think they paid a fair price? Or did they take it like they took everything else?
So I'm going to have to ask you to leave.
You got all this security in here watching me ever since I walked in, but you ain't checking for what you put in your body.
All right, mate. Let's have it. Come on, mate. Time.
I think she might not.
All right. Okay, that wahine died. The point is, we need to actually think very deeply about how a lot of artifacts, and in some cases, human remains, end up in museums in the first place, places like the British Museum where that scene took place. And when we think about the anthropology of the past, and we think about all of the studies of things like phrenology and eye color and anthropometry and hair color, we need to understand that in context of the time period. But we also need to think about what's going on now in contemporary times like today. So today, and I should warn everyone that there will be pictures included in the rest of this presentation that include physical remains, because this is something that we need to talk about and address. We can't exactly turn our heads the other direction, and these problems aren't going to go away. And that's why I am excited to talk to everyone in Hawaii about these issues. But what's going on now is very similar to what has happened in the past. So the anthropology of the present involves human remains. It involves the destruction of human remains and the extraction of DNA from those human remains and the sequencing of that DNA. So on one level, there are a lot of key insights that we can gain from this DNA about our mo'olelo, about our migratory history and our diaspora as a people. We can understand our deep past in ways that were not possible before. We can understand our deep past in context of other things like archaeology and linguistics and our oral traditions and historical traditions as Kanaka. But at the same time, there's this profound responsibility in creating an accuracy around the way that we repatriate our past using all of these different artifacts. It's back to these ideas around the beginning of archaeology, around who has, who is in control of this information and who gets to determine what the past is. And it's a profoundly complex idea. And it's not something that we're going to get to the bottom of today, but it's something that I think we should all be thinking about as we move forward. This new field is called paleogenetics. My mom likes to call it paleogenetics, but it's this idea that we are studying the past through the examination of preserved genetic material from the remains of ancient organisms. So this doesn't just extend to something like ivikupuna, it extends to other things as well. Ancient bacteria, ancient all types of creatures that have existed throughout the entirety of history, depending on how well those ancestors' remains are preserved. And it brings up a lot of interesting ethical problems around who has access, who gets access, what types of consent. Do our ancestors have consent? I would say they do, and a range of new and emerging questions. But this field is so brand new that it's sort of like the wild, wild west, sort of like frontier days here. So, and it has been transformative in terms of understanding our migratory history. If the way that we looked at the past using modern or contemporary genomic information allowed us to look at it in sort of a black and white version, ancient genomics or paleogenomics is allowing us to look at it at 1080p or 3D. It's a whole new way that we're understanding the past. We've even been able to sequence the genomes of Neanderthals that have been discovered from 50,000 years ago. It's profoundly complicated and interesting. And one of the kind of key findings that's come out of this is called introgression. And what does introgression mean? Introgression means the transfer of genetic information from one species to another as a result of hybridization between them and repeated back crossing. So what does that mean? It means that Many human beings today, modern or contemporary human beings, contain in their genomes manini fragments of Neanderthals. And this varies based on sort of your geographical history and your, your Ohana's mo'olelo, but it's very interesting to think about this as something that was previously upheld as a hypothesis, but it was proven using ancient genomic information. It's a landmark discovery that only happened in the last decade. And you can see fragments of these estimates and this sort of map of this, and this is called a heat map, and this is data from one of the first papers to come out in 2017 in the journal Science. And you can see these estimates based on individuals and where they're from and the fraction of Neanderthal ancestry that might be found in those populations. This was a very, very big deal and is continuing to be a very big deal. And even this recent estimate of 0% Neanderthal ancestry in Africa was recently disproved this year. So this field is going warp speed. All of these upheld hypotheses keep getting disproved. It's an exciting time. And that leads us to a number of new questions about things. When things happen really quickly, they're messy. They can become very sloppy. And so we have to think about this in context of what we're proving and disproving using this type of information. This brings up deep standing questions around continued desecration and exploitation of indigenous people's rights. And we get to this position and we have to ask ourselves the hard questions. Is ancient DNA research revealing new truths, or are we falling into old traps? And no one really has the answers to this yet, but I think it's fair to say that both things are true. Both things can always be true. And I want to explain a number of ways that this is impacting science and people's identity and other things. So one thing that's going on for sure is that more ancient genomes have been sequenced since 2019 than in the entirety of history. And this is research that myself and paleoanthropologist John Hawks published in the journal Nature. And we just show this upward trend, this exponential trend of more ancient genomes have been processed and sequenced in the last handful of years than in the entirety of history. And this is a profoundly complicated idea when it comes to access and how these iwi kupuna are accessed by these scientists. One thing you'll notice, this is another piece I published in the Society of American Archaeology, showing that the number of ancient DNA laboratories that require specific engineering and filtering and they're very specific labs. These labs have been on the rise since 2010. If you look at the red dots on this global map, you can see that many, many, many of these labs have been constructed all over the world. In blue, you see the labs that existed prior to 2010. Now, I should note that only a handful of labs have really been involved in this. And we have to think about what that means when a handful of scientific investigators control and dictate a lot of the information that's generated. They're processing a lot of these different remains. They have been accused of creating a paleogenomic industrial complex. And that's really complicated. Making matters even more complex is the way people acquire ancient remains. If you think that you have to go through an archaeologist, or you think that you have to go through a museum setting to acquire ancient remains, I'm here to tell you that that's not the only place that can happen. Here we have individuals bidding for all kinds of things at Christie's, and one of the most kind of sad and frustrating things that's going on today is the sale of our iwi kupuna and our ancestors' DNA online. Here, for example, is a lei made of Native Hawaiian human hair sold online for $70,000 to the highest bidder. This, to me, is very frustrating and makes me very, very angry. And it doesn't seem like Christie's and Sotheby's and other institutions are going to stop doing this unless we put pressure on them as a community. This brings up a deep series of questions around, will paleogenomics be used to further disenfranchise indigenous peoples, or is it going to empower us? And how do we see that future? So thinking about this from the point of view of Ikawa Mamua, Kawa Mahope, as we recognize and acknowledge this history of exploitation and this continued history of exploitation, I think it's high time that we think about ways to safeguard access to indigenous remains. So I'd like to highlight a few of the technologies that we're developing to prevent people from selling indigenous remains online and to sort of halt this bone rush that's happening all over the world. I was inspired by the Hope Diamond. This is a diamond that Louis XIII used to wear around his neck. What's interesting about this diamond, though, is that when it was first described, it weighed 115 carats. And then by the time it turns up at the Smithsonian century, almost a century later, it only weighs 45 carats, meaning in the process, it was stolen and pieces of it were chipped off. This got me really thinking about the way that our ancestors' indigenous remains pass hands. They're traded around like baseball cards from museum to museum. So if we can actually create systems for accountability and transparency to understand where has our ancestors' material, where have they been? Where have they been, and how long were they at X location, and how much of our ancestor did you destroy in the process? We need to understand what's going on and create transparency around that. And a very interesting thing that happened is that a Neanderthal or Denisovan pinky was described at the Max Planck Institute in Germany, and it was sent to 2 different laboratories. In the male, one of the pinky bones ends up in the lab and is processed, and that information is used to basically transform the way we think about human history. And the other half was lost in the male. Lost in the male. It's crazy. So this is the lack of transparency that's going on in this industry right now. It's happening so fast. There's this anxiety to publish in major marquee journals, and no one is really keeping a record of what's happening. When people's experiments fail, they don't report that. They only report the successes. They report when they have a new complete genome that sort of upends our understanding of history. I'm advocating for slowing down and really thinking about the ways in which we engage in this type of research. What types of partners need to be involved? How do we reach consensus? How do we involve communities in that process? And that's happening in mass at some of these laboratories as they process hundreds and thousands of different ancestors' remains from all over the world. So one of the things we've been thinking about doing is connecting CT scans and weight and other types of metrics and connecting them in a record-keeping system so we know what's being processed, when it's being processed, when there was a failed experiment, when there was an experiment that was a success, who's trading what data, and when and where and how. I think communities all over the world deserve that. The second type of technology we're exploring is something called computer vision technology. This is a very interesting new type of technology. You might have heard of artificial intelligence, AI, or machine learning technology, and I'll briefly explain how this sort of works. What we're doing is we're using images that can be sourced. So you acquire images, whether they're videos or pictures, they all include pixels, right? Then we have a second step where we analyze those images and we do something called a training data set with X number of images. And then we apply insights, score those images, and we use this process of either machine learning or deep learning to categorize those in either a what's called a supervised or unsupervised way, a biased or unbiased way. And what you can do is you can actually use these types of technologies and say, go on Instagram, for example, which is one of the largest black markets for the sales of indigenous remains on planet Earth. This isn't just a place for you to post pictures from your holiday. This is an illicit market that people are selling remains on today. And we can actually source those images, and we can find people that are involved in the illicit sales of ancient remains using artificial intelligence technology. And you can pull down the potential sales of many different types of remains. And you can even go into the chat and the way that they're communicating those sales with specific types of language. And you can geolocate these images to people that are selling different people's ancestors online, with the intent to prevent this from happening in the future. This is very much in line with a lot of the processes and procedures that we have been developing through, all over the world, thinking about enhancing ethical genomic research with indigenous communities. We will not stop doing this research. We will not allow people to continue to desecrate our ancestors, especially not online. And we've been thinking about many different ways in the future to think about disruptive and non-invasive technology. For example, this looks like a piece of or a bunch of mulch, but what this is actually is the every single attempt at trying to put together va'a that were buried in sediment prior to the development of a new technology called PEG. And what PEG allowed archaeologists all over the world to do was resuspend wood, waterlogged wood, and allow us to understand it at a totally new resolution in a non-invasive way. And here you can see one of these va'a that has been resurrected in this relief of this honu. It's remarkable, it's non-invasive, and it allows us to understand our history at a higher resolution. And that involved patience and thinking about data and archaeology in new ways. What's happening now is you have this anxiety for discovery. You have people that aren't trying to be patient enough for the development of new technologies that might involve understanding our deep past without grinding up iwi kupuna into dust. There is precedent for this. There has been the development of new technologies, something called Zooms, which involves basically taking Ziploc bags and padding the inside of the Ziploc bag and processing that collagen profile using mass spec. And they were able to determine what species of animal these Iroquois bone tips, arrow tips came from. It's pretty remarkable. Some of them came from bear, some of them came from caribou, some of them even came from human. All of that is to say, in the end of the day, we have been indigenous futurists as Kanaka, one of the first nations in the world to utilize color newspapers, one of the first nations in the world to light up Iolani Palace well before the White House. King Kamehameha himself explored putting cannons on va'a. We are not afraid of new technology. We never have been. And we will utilize the best technologies available to protect our interests, and only in ways that are supported by our community. Finally, I would just like to say that a lot of these suites of technologies and these perspectives are in light of this whole new movement of decolonial science that's going on in museums all over the world. Here we have a whole bunch of Benin bronzes from Africa that will all be repatriated within the next few years. And I just wanted to leave you guys to think about that and what the future will be like. And thank you very much. Mahalo nui loa for listening. Thanks, guys. Cool.
JoAnn, unmute.
gotta unmute, JoAnn. Aja. Okay, hope that wasn't too long. Oh, good. Right on.
Okay, sorry, I'm working off two machines and I'm clearly limited. Mahalo Keolu and Leah, thank you so much. So much to think about. And I want to look at the chat to see if there are questions coming in. Well, you got blown away by your presentation by one, so that wasn't a question, but be a nice compliment. You know, I'm going to pull up, while we're waiting for other questions, Keolu, I'm going to pull up a question that came in from our evaluation from session 1 that I think is relevant. And it says, so what does advocating for Native rights and sovereignty as they pertain to genetic information look like for geneticists, for genetic counselors, and in clinical practice too?
Serious, good question, hard question. So I think that it's multifaceted. There are so many different directions that can go. With respect to medical health, you know, I'll let Leah handle that one. But with respect to the future of the paleogenomics and how I like that paleo because it means to give voice. Yeah. So to me, it's like, it reminds me of this quote from Martin Luther King Jr. We're not made by history. We're not makers of history. We're made by history, kind of thing. It's really about allowing as many people as possible from that community to interpret information to repatriate our community and our identity today and our future. And I think a lot of the whole story and history around indigenous data sovereignty is, one, recognizing it as a resource, right? We've had that conversation last week too.
Mm-hmm.
Data itself, including digital sequence information, whether it's contemporary, modern, or ancient, is a resource. It becomes valuable. It becomes used to impute and model and predict things. And so we need to start acting like it.
And I'm going to ask that of Leah as well, because Leah, you're on the front line in clinical care. And I know that you apply this science and it is helpful with your patients and in understanding different things. So same question in that, how do we advocate for our, how do we protect that and utilize the science? And I think it comes back to your original question in part 1 is where and when do we benefit by this?
Right. So I think for us, you know, in terms of using this in our everyday clinic, I think we've certainly seen our fair share of challenges, both myself, you know, of course, myself and the genetic counselors that I work with, you know, more times than we can count, we're sitting there with families and we have this situation where we're looking at results, and these results are basically non-diagnostic or uninterpretable, or they're, you know, what they're called are variants of uncertain significance. You know, basically what it boils down to is the databases that, you know, we refer to for a lot of our medical types of genetic testing aren't really filled with people like us. And so because of that, we're at a disadvantage because, you know, and we're told, sorry, we don't know what this means. And yes, we found a change in your DNA, but we don't know if it's disease causing or if it's totally benign. And I think that it's really hard, right? Because we're the ones looking at families saying, sorry, you know, hopefully in a couple of years, maybe we'll have more information. And what that basically boils down to is health equity, you know. In the next talk, I'll put together some data that'll show some numbers as to when we have surveyed, locally here in Hawaii, the types of diagnoses that we've gotten based on, one specific kind of test, and where we have people who are admixed, and again, a lot of the Kanaka Maoli population is admixed. We don't have answers a lot of the time. And so I think, for me as a physician, I recognize that it's really beneficial to have more representation. But in order for us to do that, and I think that's another big focus, right, of our next talk, like including, we're going to address the All of Us initiative, but for us, how do we, are we cognizant of all, the different ways that, we don't want to be exploited, basically, right? We want to benefit, we want to participate, we want to be able to take a test and get an answer like any other, European would. But in order for us to do that, we have to kind of make a commitment to doing that research. One of the questions from the last talk was regarding whether or not, we had the ability to kind of do a completely indigenous run, right? Biogenomic repository for Native Hawaiians. And I think that's certainly a question. Right now we're kind of laying the foundation and kind of educating the community. And I think looking forward, we need to think about, what Hawaiians as a whole want. And hopefully that's health equity as well.
Yes, thanks. Listen, I've got a great question from Dr. Francine Gachipin, who has been a real awesome mentor to us as we've looked into genetic studies through Imihale in the past. She is from Arizona, but and I know she's a colleague of yours, Keolu. But her question was, as a regular person, how can we help stop the sale of our artifacts on social media and auction houses?
Yeah, that's a great question. Wonderful to see you here too. It's always awesome. I think, like, first and foremost, you don't have to sit here and design algorithms like us. Like, maybe that's not the best use of your time. But you can report those people if you see anything like that. This is a human rights violation. It doesn't matter what community you're from. We would never go to Arlington Cemetery and dig up someone's ancestors. It's wrong. And so that's one way. And I think the second way is just bringing this up as a larger issue with your community, with your ohana when you're, having dinner or beers or whatever, but making sure that people understand that this is happening. And, sourcing and understanding what people want. And I think that most people don't want that to happen.
Yes, I also think, some of us who are in the session tonight, have some experience with research, with institutional review boards, with the kinds of mechanisms that are supposed to protect technically. But we know that that's complicated too. It takes decades sometimes for science to correct itself when something egregious is going on. So we have so many case studies about that. But again, looking at informed consent, and I think that's something you both are going to touch upon in session 3. But, there are some examples. At Imihale, we work with the Queen's Medical Center that actually allows patients who have remnant tissue after cancer surgery to be reconsented if that's their choice. And we know that in developing that policy with the Queen's Medical Center, that it was, one, quite novel. Very few hospitals do it. Most people sign away access to their tissue during surgery as part of the general consent. But in fact, they did revise their protocols about, I want to say 6 years ago now, for their biospecimen bank, that if the patient wants to be reconsented, they can be. So I know there are steps to look at transparency and equity and informed consent, but clearly not enough. And as I said, quoting your opening paragraph, Keolu, in your publication, is the history is so dismal that, we have to start to work through that and hopefully learn from that. Yeah. But which is part of the reason behind these webinars. Emmet, you have Any questions too?
So I'm just made aware of a patient who's being treated for breast cancer, and the folks taking care of her is a group working as genomic health. I
Mm.
mean, that's how they kind of like advertise and in the reports that I've been forwarded, genomic health. And I'm just saying, wow, it's the first time I've heard. What is genomic health?
Great question. I think that certainly is a nice umbrella term, but when we're thinking about genomic health, right, it's using our DNA to kind of guide and give some perspective on in terms of, individualized or personalized medicine. So in regards to your patient, Dr. Aluli, would for her diagnosis of breast cancer, we probably looked at a panel of several dozen genes to try to identify, right, which one had the misspelling that was pathogenic or known to cause, right, or put her at increased risk to develop, again, that tumor. So that is one way, right, looking at disease and risk, or trying to hone in on a diagnosis. Another up and coming area of genomic health is, within the realm of personalized medicine, is pharmacogenomics, right? So basically looking at your genomic information to see how you might metabolize a specific medicine. People who are on, blood thinners, or people who are taking different types of, heart medicines, how is your body going to react to that? How is your liver going to clear that? Is it going to break it down and flush it out too fast, too slow? And, what are, again, the consequences related to, again, those underlying variations in your DNA when it comes to taking medicine? So I think when we're talking about genomic health, we're thinking about, not just making diagnoses, but also too how we can maybe, I guess, make medicine more personal too.
I have, thank you, Leah. I have three questions, but I'm going to start. One is for you, Keolu. How are you going to handle bone fragments in your training data set? Larger bones seem like they would be difficult enough, but small bones, all way.
That's an interesting question. Before I answer that, I just want to piggyback on the question about genomic health too.
Yes,
And
please.
just like kind of preview next week a little too.
Yeah.
We're
Yes,
gonna
please.
talk about weird. I mean, there's a whole new emerging, fascinating whole discipline of gene therapy that is emerging. And I think this is a huge area that people should keep their eye on in understanding the potential for genome editing and what that means in context of removing pathogenic mutations in the first place, and what the sort of ethical dilemmas are there too. So that'll be fun for us to talk about next week. But also thinking about this idea of, so I want people to understand in terms of bone fragments and sizes of different bones and so on and so forth. This is a good question. Certain parts of the human skeleton yield higher concentration of DNA, and this is one of the major problems, is that these predatory scientists are hunting after skeletons and skulls for a reason, right? Skulls contain something called the petrous bone, the bone that is wrapped up in like a little onion, and inside of that little onion or gyroscope is a bone fragment that contains like 100 times higher DNA concentration than anywhere else in the skeleton, right? So this means that they're going to prioritize that specific bone. So that's something to think about. So this is a good question, but I think most people in this industry are motivated by that, if that's helpful.
Okay, thanks, Keolu. Here's another question by Dustin. Is investigation into DNA extracted from Hawaiian ancestors already underway, or are we possibly close to that happening? If so, who is doing this research, and what avenues do we have for the Hawaiian community to get involved in directing these research objectives? More importantly, What avenues do we have for the Native Hawaiian community to direct matters of consent, to be able to say yes or no to doing this research, and also dictate how?
Want to go first? Yeah. Oh, I mean, I can tell you, everybody, this is happening, very much so, and you need to voice your opinion and stand up for our community's indigenous data sovereignty. This is happening. I mean, you want names? No, but but I think that there's a much larger conversation that needs to happen with all types of different stakeholders from our community. And I think whoever asked that question, that's an excellent question that I hope everyone is thinking about.
And I would recommend for Justin to look at part 1 if you have not, because I think what was very insightful and disturbing was the sale of access to the 23andMe and Ancestry.com samples. So a lot of it is in the name of future research. But again, who has control over future research? That doesn't tell us who, how, or why. So I do recommend you listen to part 1. And then, of course, part 3 is going to be much more to the point and present. So Lehua has a question. Is genetic testing encouraged without a specific medical concern?
Great question, Lehua, and aloha. Sorry, that's actually, I didn't see Keolu making signs at me because I was reading Lehua's question. And this is a great one. A long time no talk. Lehua and her family grew up in Haiti as well. So we were neighbors, childhood friends. Anyway, Lehua, I think that is the perfect question. And when we're talking about, genetic testing, I would say that it is definitely not encouraged without a specific medical concern. So a lot of what I guess is important to understand, or I guess the crux of looking at DNA, is what we call genotype phenotype correlation, meaning, looking at the DNA and then understanding what that protein does in the body, where it's expressed in different tissues, and understanding at like a cellular and molecular level what that means, and then using that to then translate into, do we see that in the patient, and is that make sense? And so without actually having a medical concern and then going out and getting genetic testing done, it makes it really, really difficult, right? So when we see any patient in our office, and we don't recommend testing for things that are uncertain. And again, if you don't have a symptom, we don't recommend testing. So let's say, for example, again, we've talked about breast cancer a lot, but it's, pretty common in the, Native Hawaiian community. So hereditary breast and ovarian cancer. So maybe you come to our office and you have not been diagnosed as a patient, but let's say, maybe my grandma and her sister and, my cousins who are older than me, they've been diagnosed with, either hereditary breast or ovarian cancer, and they were, in their 30s or in their 40s, which is kind of, a younger age. And so we may not, with that history, we might not do testing for you per se, but we would then be able to recommend testing for an affected individual, because it's much more powerful to look at that gene difference in somebody who actually has the tumor or the breast cancer to make sense of it. Because inevitably, like we had mentioned before, the databases don't have a lot of people like us who are admixed. And so the challenge then becomes to kind of sift out what is real and what is not, because a lot of it is going to be uncertain, especially when, you're of indigenous descent.
So Leah, to continue on that, the next question, and you spoke about it a little bit in part 1. What are your thoughts on DNA testing at companies such as Ancestry.com or 23andMe, MyHeritage, Family Tree DNA? And so Elena, who forwarded this question, it was addressed a little bit in part 1. It's specifically with the BRCA1. So Leah, can you expound a little bit on that?
Yeah. So I guess sorry, one more time, JoAnn, just to make sure I got the question right.
Oh, hold on. Let me open it back up. It says, what are your thoughts on DNA testing at companies such as Ancestry.com, 23?
Yeah, okay, got it. You know, at least for the ancestry portion, you know, it's certainly like a fun exercise that may be interesting, right, to do with your family or with other, you know, for other purposes, you know, but also to remember that when you're doing that, you might be getting answers that you weren't expecting, right? Ancestry testing can tell you, you know, whether your mom is your mom or your dad is your dad, and people don't really, you know, kind of think about that before they click through and just say, yeah, I consent and use my DNA. I think it can certainly be somewhat representative, right? In terms of the databases, they're pretty good in terms of using, you know, a global population to kind of narrow down, but they won't be able to say, yeah, you are, you know, Native Hawaiian, or you're 5% Hawaiian and 15% Tahitian, right? It's more like, because we're so much migration very recently, we're not that divergent genetically. And so that's why we're kind of grouped as Polynesians on a lot of these tests. So I think it's, you know, okay. I would not recommend these tests for health reasons. And again, they are not certified. You know, for medical genetics, we actually use specific labs that have special certifications that, you know, actually are upheld for industry standards throughout, you know, this country and many other countries. And, you know, again, the ancestry, the 23andMe, the things that are over the counter are not certified to be, you know, for health.
And I think the cautionary thing too is that, again, they just sold access to their samples. So I took it because my kids gave it to me as a Christmas present, but I was not told it would be open grabs after, right? And so, and you may think, well, but they won't know it's my saliva or whatever. But let's face it, so much of bad science has extrapolated things on Native Hawaiians that are not true. And this is what in review boards and in science is called group harm. And this is where stereotypes and negative stereotypes in particular get perpetuated based on small or bad science. So just keep that in mind. Don't buy it for Christmas for your friends, or let them know it might be up for grabs in the next sale.
Right. Again, you know, we talk about all those examples for pharmacogenomics, right? And they send it, you know, off to make some super drug. And, you know, you always hear about that story about how they caught that serial killer on the mainland by looking at these databases. And, you know, I mean, really, people are not really thinking it through. And so just to be aware, and if that's your choice, again, for ancestry purposes, I think, you know, that's certainly got some validation, but not for medical.
Could I add to that as well? Just the idea that, okay, just here are the facts as we're all alluding to. 23andMe sold access to their database, potentially including your genomic information, to GlaxoSmithKline for $300 million. This year, they created their first blockbuster drug in consort with a pharmaceutical company in Spain, and they're going to probably sell that back to you. And you're not going to get subsidized access to the drug. You're not going to get a percentage of the profits. You're not going to get a percentage of the intellectual property, even though they very well could have used your genome to derive and refine the biomarker that developed the drug. So, and this is something we'll get to next week as well. But I think that that's something we need to be conscious of. Like, at what point is you participating in their thing and paying them $150 actually helping you with anything if your parents already told you where you're from?
Agreed. Thank you, Keolu. I'm sorry, I'm looking back at the chat and I can see that Elena's initial question about ancestry testing and over-the-counter things was more pertaining to if you're adopted and if you don't know your family. You know, again, you know, knowing kind of where your, you know, ancestral roots came from, that would be certainly appropriate. But again, thinking about that database question, you know, if you were adopted and again, you don't know who your family is per se, the only way that you would get a match even for, you know, second or even third degree relatives, right? Second cousins, third cousins, that kind of thing, or even, you know, parent or half sibling would be if they've also participated in sharing their DNA within that same company.
Great point.
Okay, so, you know, we're coming close to our time, and I just wanted to ask both our panelists, first, thank you so much. But if you had any last thoughts, maybe a little teaser for next week. or not next week, for the next session, and we will let you know when that is. Keolu?
Just mahalo nui for everyone who came. It's always such an honor and privilege to get to talk to our community about all of these issues and let you know what's happening now, in the past, and moving into the future. And we're so excited to talk to you about all of these new developments next time.
Thank
Right.
you, Leah.
Yeah, of course, piggybacking on that, again, so grateful to be able to share what, we are so passionate about, genetics and genomics. And again, we're so excited to have all this feedback and participation. We really do appreciate all the thought and the thought-provoking questions that you guys send back. Just again, to lay the foundation, we are trying to just educate the community at large so that we can have better discussions and hopefully get more input, not just from our own indigenous community, but other indigenous communities as well, on how to move forward with, doing smart science, right? Again, there's so much that we can benefit from genomics, but we also have to make sure that we're doing it in a pono way.
Yes. And part of this whole education push is because there is a large study and it's right on our door and it's in our island. And so let's make informed decisions. Let's figure out what else we need to know, what other questions we need to ask, what other potential there is so that we can address the things that have come up at both sessions and in the literature. And that is involvement, transparency, equity in resources, equity in access. And as we say, who really benefits? I'm going to let Emmet close off this session with his wise words and magic.
So this is really exciting. It's really kind of something that we really need to pay attention to. And I'm really thankful that you're bringing everything out and balancing everything and keeping the Lahui, the Pa'aina, the kind of like Pacific physicians involved. I just talked with Chair of Office of Hawaiian Affairs, Clint Machado, and she was just really exciting to have OHA involved in some way. And so there'll be ongoing talks and ongoing ways we can gather as those who have been educated by these webinars, and hopefully we'll be able to make some progress and be able to say, this is what we need as Lahui, Hawaii. And thanks, guys. And JoAnn,
Thanks,
great.
Emmet. Well,
And
and I just want to let everybody know there's a evaluation that follows this, so please fill it out. It gave us a lot of good input and questions for the upcoming sessions. And also thanking our sponsors, Papa Ola Lōkahi, Office of Hawaiian Affairs, and we're looking forward to including Ahahui as a sponsor, and also Ahakane. So thank you, everybody. Thank you to our speakers. Awesome as usual. Mahalo, Emmet.
mahalo, JoAnn. Mahalo, Kim.
Thank you, Kim and staff, for doing the behind the scenes. Yeah. Okay. Yay.
Aloha.
Aloha, Keolu and Leah. Thank you so much.
Malama, malama.
Can I get out of here? I don't know how to. There we go. Okay. Oops.